this scientific research opportunity to research more on the field of pharmacy, but in particular pharmacogenomics. So I started by going in the past and just finding out more about where this field started. So the pharmacy comes from the Greek word pharmakon, and from my research I found out that the field of pharmacy the modern field started basically in Europe and spread out in East Asia, and then after that moved on to the rest of the world. So some of the oldest pharmacies are in Europe, such as this one here called the Golden Pharmacy, which is in Trier, Germany, and it opened in 1241. So pharmacognosy is basically what the earliest pharmacists practiced, and it's basically just using the natural world, so roots, plants, trees, to make drugs, to help people through diseases. So pretty much it wasn't very effective because you didn't know that one concentration of something is going to be the same as the next for the next person. So now with sophisticated technology, people are able to synthesize compounds that are very detailed and will be able to help people better. So studies show that, or it's estimated that 100,000 Americans die from drugs, like adverse drug reactions, and it's a bit ironic because something you're taking to prolong your life or to help you can actually kill you. So even if it doesn't kill you, there are certain side effects that come from taking drugs, like you become dizzy, you can become blind, even, you're taking a drug to cure another disease, but you get another disease from taking that drug through liver failure, heart failure, and so on. So what can be done to make drugs, I guess, more efficient for you? So this comes into pharmacogenomics, which is the use of your DNA to manufacture drugs that are specific to your body. So I won't go, well, Lou did me a favor and explained a bit about the DNA structure. So DNA holds the coding for genes which manufacture proteins. Now, each of us have basically the same type of things in our bodies, tissues and bones and everything, but then our DNA, there's slight differences between each person. So I may make the same protein, but then it'll be a bit different from the protein that Ms. Walter can make. So let me give an example of how this can affect drugs. There's a drug called pravastatine, which is used to lower your cholesterol, but people that have an enzyme called cholesterol transfer protein, like a different kind of that enzyme, the drug won't be able to work for them, so it won't lower their cholesterol. So with pharmacogenomics, you look at your DNA, and then after that, there are different regions in the DNA that are different, as I was saying, and they're called, sorry, let me just check. They're different, they're named differently, but then for this specific, it's called single nucleotide polymorphism. And so scientists will be able to look at these regions and then associate them to different drug responses, and then through that, if you were to take a genetic screening and then find that you have a certain single nucleotide polymorphism, they'll be able to determine which drug would work for you and which wouldn't. So when you talk about Africa and pharmacogenomics, does it have potential? I think so. There have been studies by the University of Nairobi and Oxford University where they were able to make a DNA vaccine for AIDS. It's in its clinical trials now, and I'm not sure how successful it has been. But this just shows that with genetics, we'll maybe be able to solve some of the diseases in Africa that plague us a lot, like malaria, tuberculosis, and HIV. So I'm optimistic that DNA and that study will be able to help Africa move forward in the health field.
Thank you very much, Bridget. Okay, questions for Bridget?
So what's the difference between pharmacogenomics and gene therapy?
Well, from what we were saying, gene therapy is you're taking a gene, like you have someone has their DNA and there's a faulty gene or something that's making them sick, so you take another gene and insert it there so that am I saying it right? So, but then with pharmacogenomics, you look at the DNA and you find the regions in the DNA that are different and that could cause an adverse drug reaction, and therefore you make it so that the person doesn't take the drugs that will cause them to die or get sick or so forth.
How does this work in terms of the HIV sort of treatment? Is it that do some HIV treatments only work for certain people?
I'm not really sure because the study, it didn't tell exactly how they did the vaccine or how, but they were looking at people, especially prostitutes in Nairobi who were resistant to it or something, I think, and then used their DNA to make the vaccine. But
okay, yeah.
Yeah, so I don't know the details of that though.
Yeah. Therapeutics.
How are they going to make the vaccine for malaria or to cure malaria?
Well, that's the thing. They don't know. They haven't done that. They haven't actually started that yet. But I'm saying that if we can, like we could work with DNA, you and I, and then we can make drugs that either stop malaria or you could do the gene therapies somehow in the course of the malaria cycle.
Excellent. Thank you very much for